So the movement of the bacteria, different bacteria move in the different ways by use of the flagella or the cilia. Flagella is the legs of the bacteria and cilia are the slender hair-like, whip-like structure by which the bacteria can swim and attach to the different surfaces. Flagella are longer and cilia are shorter. These enable bacteria to move in a liquid called locomotion by these cilia. These are just the oars of the ship of bacteria. Flagella move in a snake-like movement and cilia move in a rowing motion. Self-movement motility as in the spirochetes or the spirillea by the gliding movement, so they can have a bit movement which is called the gliding movement. The cocci bacteria are transmitted in air, in dust, or in the substance where they settle. They don't have any motility or self-movement, but they can have some gliding movement like the twist, so they can move from one place to another. So one can see the flagella and the cilia here. These cilia are outside the cell wall and the flagellum that rise within the body of the cell wall. So that is meant for the locomotion and this is meant for just some swimming and attachment. Now, bacterial reproduction, how the bacteria do produce and divide. There are 5 ways by which bacteria reproduce: binary fission, conjugation, sporulation, budding, and fragmentation. Asexual reproduction is the binary fission. Single parent cell divide to form 2 daughter cells. Single DNA molecule replicates, each copy attached to the cell membrane. Cell begins to pull apart and produce 2 cells of the identical genetic composition that have been transmitted to the each daughter cell. Then conjugation, it's a type of the bacterial sex, occurs only between closely related bacterial strains. 2 organisms embrace and share genetic information such as resistance to the penicillin. Here one can see the binary fission, the cell divide and enlarges and then it becomes 2 and having the genetic material transferred to that. So here you can see the example of binary fission. Here bacterial conjugation, the bacterial sex. This bacteria, they do have attachment with the pilus and it just have just transmission of the some genetic material and ultimately those genetic material is transferred from one to the another and these bacteria become the new donor and they are capable of just giving this donation to other bacteria. So it's a kind of bacterial sex. Then sporulation, spore formation. Bacteria can survive unfavorable condition by producing an endospore. These are highly resistant structure formed in extreme condition when nutrients are depleted. So bacteria become endospore which can resist all these harsh and extreme environment seen only in gram-positive spore-forming bacillus species like Bacillus cereus, anthrax, tetanus, and botulism. Spore contains bacterial DNA, cytoplasm, some membrane, peptidoglycan, water, and keratin-like coat. Coat is resistant to the heat, dehydration, radiation, and chemical. That's why the spore is able to resist the harsh environment and can survive even this bad and harsh environment. It is formed by the dipicolinic acid, a calcium ion chelator, which is resistant coat, which cannot be just pierced by the different conditions of the environment. Here one can see the sporulation, the vegetative cell due to unfavorable condition just goes into this structure like this. At one end there is a spore formation and this spore formation is able to resist the extreme environment, and spore is just going to detach from the mother cell, and this free endospore is able to live independently till the time it gets the proper nutrition, favorable condition, and then again it can become vegetative cell which is capable of dividing. Spore is not able to divide, and it can live to transform into the vegetative cell which is capable of dividing. Here one can see the structure of the spore, that is the core and cell wall, outer membrane, and the coat which is very resistant to the outside world and impermeable to the outside world. Spore formation, after formation, spore become just idle, have no activity, remain dormant for years and years in the soil, and wound contamination, as one have heard of the tetanus, has spores of the tetanus and the gas gangrene. When exposure to this suitable nutrition, these spores get entry to the human body. Specific enzymes degrade the coat of the spores. Spores germinate into the pathogenic vegetative bacterial cells. One bacterial cell produces one spore that germinate into one cell, and sterilization by the autoclave only at 120 degree centigrade for 30 minutes can only kill the spores and cause sterility, or only sporicidal chemical disinfectants will kill spores. Antibiotics are ineffective because spore wall does not allow them, and that is impermeable to them, so they can't reach and cause the destruction of the spore. Here one can see vegetative cell changes into the spore and detaches from the mother cell. Now this spore, on export to the suitable environment, just changes to the vegetative cell and able to divide. Then there is a kind of bacterial reproduction, is the budding. Certain bacteria like Mycobacteria, Corynebacterium, and Hyphomicrobium divide by budding. The budding cell originates as an outgrowth of the original or the mother cell, which eventually reaches to the size of the mother cell, mean the detached or outgrowth of the mother cell is capable of making the new cell. The bud now separate to have independent resistor, as it is in case of the yeast. Fragmentation, several actinomyces form filamentous cell. These reproduce by simple fragmentation of the original filaments into the new cell. So this process in which the fragment is capable of producing the new cell is called fragmentation. Here one can see the outgrowth as a bud that detaches from the mother cell to become the new independent cell, and this is the fragmentation. One can see the piece of the cell detaches from the mother cell, and that is capable of producing the new cell. Now, pathogenesis of the bacterial infection. The disease caused by the bacteria, local or the systemic, is due to the production of the toxins, hypersensitivity reaction in the body, and then hematological or lymphatic dissemination when the bacteria get entry into the blood or the lymphatic vessel to reach to the different organs and causing the infection there. So number one is the production of the toxins. Many bacteria produce harmful compounds which are called toxins. Bacterial toxins are of two types, exotoxin and endotoxin. Their name tells from where they do come. So sight is what you see with your eyes, and vision is what you see with your mind. So endotoxin are produced by the living bacteria, and the endotoxin are produced when a bacteria do die. Source of the exotoxin is the gram-positive bacteria and the gram-negative bacteria. But in case of the endotoxin, it is only the cell wall of the gram-negative bacteria when the bacteria die and that behave as endotoxin. These are secreted from the bacteria, exotoxin, and these are not secreted by the bacteria. Chemistry of the exotoxin is polypeptide, and these are, in case of the endotoxin, is the lipopolysaccharide, which is abbreviated as LPS. These are highly potent exotoxin, extremely lethal, and these are less potent and low potency as well. These are thermolabile, destroyed by the heat. These are heat resistant. These are neutralized by the specific antibodies. If some antitoxin is given, that can just neutralize the exotoxin, but these are not neutralized by the specific antibodies. Vaccine toxides are available against exotoxin. These are not available in case of endotoxin. Clostridium tetani and other diphtheria are the bacteria which release exotoxin, and the Salmonella typhi and many gram-negative rods. These are going to produce the exotoxin. Here one can see the cell wall, the bacteria is intact, and from the inside it is going to release the exotoxin, which are going to act at somewhere else. These are just carried away by the blood or by the lymphatic to reach to the different organs. And here, in case of the gram-negative bacteria, one can see the cell wall. When the bacteria dies, the cell wall fragmentation, these act as the endotoxin. So here again one can see the exotoxin. These are just released by the, and these are the cell wall of the gram-negative bacteria, which is having three parts, and that totally behave like exotoxin. Then hypersensitivity reaction, it is a reaction between the bacterial protein and the sensitized lymphocytes initiate inflammatory process in that part where the bacteria do lodge. Then hematological or lymphatic dissemination, as I have told you, the bacteria get entry into the blood or the lymphatics, so the involvement of the distant sites through this dissemination or spread from a primary focus, bacteria travel through the bloodstream, causing bacteria or to reach to the lymph nodes. So that is all about the bacterial different pathogenesis and about the bacterial cytology and different function of the bacteria. So if you have liked the video, please subscribe it, or if you don't like, please let me know for any. Don't hesitate to say anything about. My numbers are here, and thank you very much for watching.